All right, so I want to welcome everyone back. This is going to be our screencast for Chapter 15. What's really nice about 15 is that a lot of the information that we had learned back in Chapter 14 is going to have a really strong connection to what we're going to look at brand new in Chapter 15. The idea of inheritance, of course, and we're going to look at some of the concepts of Mendelian inheritance. But what we're going to do is we're now going to look at the structure of the chromosome and basically try to understand how the structure of the chromosome, the behavior of the chromosome, is going to influence the passing of those traits onto the next generation. So before we get too far into 15, we want to go ahead and look at, again, the simple idea of Mendelian inheritance. Now, something we're going to focus on at the very beginning is something called the chromosome theory of inheritance. If you look here, we have 2 bullet points. It says Mendelian genes have specific loci. Remember, loci are simply the position of that gene on the chromosomes, and the idea that chromosomes do undergo something called segregation. So remember, segregation is simply the separation of those chromosomes into gametes when those gametes are being formed. So the separation of those homologous pairs is what we're looking at. And of course, the idea of independent assortment. So thinking about that typical 9:3 to 3:1 ratio that we had looked at with the dihybrid cross, that demonstrates the idea of independent assortment. In other words, we have maybe 2 traits, which we do in a dihybrid cross, but they really don't influence the passing into those gametes. In other words, they're not carried together, and so that would be considered independent. So one does not affect the other. Now, the behavior of the chromosomes during meiosis was said to account for Mendel's laws of segregation and independent assortment. And so I have a diagram over here of meiosis, and this is something that you had looked at back in Chapter 13. What they've done here is they've connected up meiosis with a particular set of traits, and they're looking at the color of the seed, and they're also looking at the texture of that seed. So whether it would be a smooth, round seed or a wrinkled seed. And so we simply have 2 different traits we're going to follow through. So remember, in the P generation, that's going to be our true breeding parents, and so they're homozygous for both of those traits. When you look at the gametes that are being formed by each of those, there's not a lot of variety in the gametes. In other words, they can only pass on, in this case, if there's 2 big Y's and 2 big R's, they can only pass on a big Y and a big R. So that's all their gametes will have. And the same thing goes for the homozygous recessive parent that you see in the upper right. And so when those gametes are produced, those are going to create our F1 generation. So those are the offspring that are produced from the parental generation. And if you think about it, when you look at, again, we're going to connect up meiosis to this, you got to think about the gametes that can be produced from those F1 individuals. And so if we do a cross with that F1 generation, the idea now is that they are heterozygous for both traits. Now, we know that yellow is dominant over green, and round is going to be dominant, or smooth is dominant over wrinkled. And so both of these offspring that are produced using those gametes are going to have gametes or produce gametes that are going to have one of each of the different alleles that are responsible for those 2 traits. Now, when you look down here at metaphase 1, remember during metaphase 1, we basically have the lining up of those duplicated homologous pairs. And if you notice over here on the right, we have a little bit of a difference in regards to right there. You have, instead of a solid blue line and a solid red line, what we have is down here, we have, it looks like they sort of switched places with those chromatids. So remember, during meiosis 1, we have something called crossing over. Now be aware that crossing over actually occurs during prophase 1, but it's being represented as being complete in metaphase 1. Now the reason that becomes important is because later on in the screencast we're going to talk about how that crossing over is going to influence what you would see in your offspring. But in this case, what we're looking at is simply the idea of the separation of those homologous pairs. And so if you notice, they do pull to each side of the cell. And then eventually during meiosis 2, we have those sister chromatids for each of those homologous pairs that are also going to separate. And so when they do that, these would be the gametes that you would get from that particular meiotic event. And so when you think about all of the different varieties of seeds that would be produced at the very end, based on the gametes that are available from this original pair, that's what's going to give us that 9 to 3 to 3 to 1. So basically every combination that would be possible from this particular cross is going to be represented in that 16-box Punnett square. And that's what we mean by independent assortment. So we have one gene not influencing another gene. And again, typically when you talk about typical Mendelian inheritance, these would be genes that would be on different chromosomes. So then what we have to do next is we have to think about, so what does that do in terms of inheritance and what we're trying to learn in chapter 15. So basically when you think about the inheritance, sometimes people sort of think Mendelian inheritance is pretty straightforward and there can be no deviation from the typical inheritance that we would think about mathematically. But somebody by the name of Thomas Hunt Morgan, he was an embryologist, he actually discovered the evidence that would support that sometimes we have genes that actually are on the same chromosome. And so what he did is he did some experiments with fruit flies, and you guys did something similar to this in your fruit fly simulation on the computer. These particular experiments he did with fruit flies provided the evidence that he needed that chromosomes, well, first, are basically the location for these heritable factors. In other words, the genes are located on chromosomes. But there was one experiment that he did where he mated male fruit flies with white eyes, and this is considered a mutant, with female flies with red eyes. And so this would be the traditional wild type fly. And so you can see the cross right up here. And so this would be considered the P generation. Now, when you look at the F generation, if you notice, all the offspring had red eyes. And so that would be your typical, what we would expect, dominant over recessive idea is that that generation is going to just have that one characteristic. But if you look down here for the results of the F2 generation, that's where things got a little bit different. If you notice, we have basically a 3-to-1 ratio. And the reason that becomes a little bit different is because you notice that we have little symbols next to each fly, and that little symbol is representing whether or not that fly is male or female. And so if you notice on the left, the females all had red eyes, and in this case the males, we actually had 50% that had red and actually 50% that had white eyes, so that recessive trait. And so what was kind of unusual here is that none of the females showed up with that white-eyed trait. Now, so if you look down here, what he did is he decided to go ahead and kind of think about where these genes are located. And what he did is he determined that the wide-eyed mutant allele had to be located on the X chromosome. Now remember, we had talked about sex chromosomes back in chapter 14. Typically when you think about sex chromosomes for some animals, for us for example, females are going to be XX and males are going to be XY. He reasoned that this particular gene must be a gene that's going to be located on those sex chromosomes. So if you notice, based on what he had seen, the P generation, and if you look over here, this would be the female. The female has 2 X's. Each gene that is going to be represented in that female was the wild type gene. Now, of course, in the male, again, this is the P generation, so they're both true breeding. This male was wide-eyed, so he had the recessive gene. Now, because he has a Y chromosome, very few genes are found on the Y chromosome. So he only has one copy of that recessive gene. So the only thing he can actually pass on to his offspring are going to be either that X with the lowercase w, or he's going to pass on that Y chromosome. Now, if you notice for the female, she's only going to pass on, again, that wild type gene because she has red eyes, and again, this is the P generation. So this would be considered the F1 generation of the cross. And so if you notice from the flies that are being produced, all females are going to have red eyes, and all males are going to have red eyes simply because the female is going to contribute the X and the male is going to contribute the Y chromosome to their male offspring. But if you look at this F1 generation, if you take 2 of those heterozygous individuals and cross them, that's where things get a little bit different than you would find in typical Mendelian genetics. If you notice down here in the Punnett square, the female can actually produce either a wild type or the wide-eyed gene because she's heterozygous for that gene. The male can only contribute the wild type because remember, he only has one copy of that gene. So when you put that information into a Punnett square, what you end up with is you end up with, again, 50% of those, or actually all of the females have the red eyes. But again, you have that sort of unusual situation where you have 50% of the males with red and 50% of the males actually having white eyes. So that actually supported the idea of the chromosome theory of inheritance, the idea that these genes are located on chromosomes. So based on the information produced by Morgan, now what we need to think about is we need to think about the idea of sex-linked genes. And we actually were introduced to this back in chapter 14 because you guys did some Punnett squares regarding this particular concept. So in humans and again in some other animals, again like we had said before in the previous screen, there is a chromosomal basis of sex determination. In other words, we're talking about basically them having a certain arrangement of chromosomes which would indicate whether or not they are male or female. There are typically, for these animals, two varieties of sex chromosomes. Remember we had said there could be an X and there could be a Y, so very different chromosomes. And it says only on the ends of the Y chromosomes would there be regions that could possibly be homologous with the X chromosome. The reason for that is because the Y chromosome first is a little bit of a different shape and it's usually typically much, much smaller. Now there is a particular gene that's really important on that Y chromosome and it's called the SRY gene. And that gene is so important because what it does is it actually codes for the development of the testes in the male. Now as we had said, females typically tend to be XX and the representation for males is going to be XY. So each ovum or sex cell is going to contain an X chromosome while a sperm may contain either an X or a Y. Now a gene located on either sex chromosome, as we had sort of pointed out both in chapter 14 and what we're doing right now, is called a sex-linked gene. Now when you have sex-linked genes in mammals, typically one of the two X chromosomes that you would find in that female is going to be randomly inactivated. In other words, it's going to be turned off during embryonic development. Now, when that occurs, this inactive X is going to actually condense or become smaller, and we're going to refer to it as a bar body. So if a female is heterozygous for a particular gene located on that X chromosome, she will be considered a mosaic for whatever character is located on that X. Cats are a really good example to sort of illustrate this idea of a mosaic pattern. So over here again, if you think about the early embryo, when you think about the X chromosomes, remember they have two X's. There could be an allele for orange fur, so we're talking about fur color, and then of course an allele for black fur. So cell division will occur and X chromosome inactivation will happen. The thing about this whole idea of X chromosome inactivation kind of throws something else into this whole mix, is because sometimes the cells that you would find in that particular animal, different cells can have different inactive X chromosomes. So in other words, what that means is you get this color pattern simply because if you look at the diagram, again, they have two X's, but maybe for this particular part of the body, the black fur is being represented. In other words, we have inactivated the orange colored gene, so all of the cells in this particular part of the body are going to have black fur, while over here in this part of the animal's body, we have inactivated the black color gene and have activated the orange color gene. So that's what they mean by the idea of a mosaic, or basically different cell populations in that one animal, basically expressing different phenotypic traits for that particular gene. Now, linked genes tend to be inherited together. So we're going to continue the idea of these linked genes, but not necessarily sex-linked. They can also be autosomal as well. But I want you to keep in mind that they do tend to be inherited together because they are located near each other on the same chromosome. So basically, through another series of experiments, Morgan had found that body color and wing size are actually inherited together based on the numbers that he had found in his experiments. And when they're inherited together, they tend to produce very specific combinations. And we refer to those combinations as being parental phenotypes. In other words, we have offspring that resemble the parents. So over here on the right, what we have is, again, just another typical cross. This is going to be considered our P generation, so these animals are going to be homozygous for each of the different characteristics that we're looking at. This is your typical wild type or gray-bodied, normal-winged individual. and we're going to cross it with, again, it's the P generation, a true breeding double mutant. So it's going to have a black body, and the wings are going to be a bit different. They're going to be considered vestigial wings, or very small wings. So when we make the cross, again, we get a dihybrid, so we get the wild type that's going to be basically heterozygous for each of those traits. But remember, we have basically a dominant over a recessive type of condition here. So if you look at what is produced, we basically have animals, and in this case we have a female, that basically have that wild type representation for body color and the wings. What we're going to do next is we're actually going to do a test cross. And remember, when we do a test cross, we're going to cross that individual with a double mutant. So we're going to cross it with an individual that actually is recessive for both of those traits. When you do the test cross, what you would expect is you would expect this type of ratios. So we have the male being represented here, again, recessive for both, and we have the heterozygous female, in other words, heterozygous for both traits. Her gametes are being represented here. So you would expect a 1:1:1 ratio. But if the genes are located on the same chromosome, in other words, they're linked, then you would expect a 1:1 and 0:0 type of ratio. All right? But based on the results that he had found, that's not actually what occurred. What he found is, based on the numbers that he had seen, he actually did get some representation of both of these other phenotypes that you would see in the fly. So he didn't get the second one. He didn't get the 50-50-50-50, or I guess it's not that, it's 25-25-25-25. He actually ended up with something a little bit different. So, however, as we can see in the diagram, non-parental phenotypes were also produced. Now, when you get the production of these non-parental phenotypes, we're going to give them a special name. We're going to call them the recombinant types. And what this indicates is that some genetic recombination had occurred during meiosis, probably, and had actually produced those other two phenotypes that we did not expect. So basically, based on the information that Morgan had found, he had discovered that genes can be linked, but the linkage itself was actually incomplete, and this was evident from the recombinant phenotypes that he had seen. Morgan proposed that some process must sometimes break the physical connection between genes on the same chromosome. Now, this is actually something we had looked at, again, like I had said earlier in the screencast, back in Chapter 13. When you guys had done your study of meiosis, remember that we had talked about a process called crossing over between homologous chromosomes. And so he theorized that this was probably the situation that was occurring, or that was basically giving us the results that he was seeing in the number of flies and different varieties of flies that were being produced. So again, looking back up here, again, this is going to represent that test cross that we had looked at in the previous screen. And so if you notice, the idea is that when you cross it with a recessive individual, remember, the only gametes that can actually be produced during that time are going to be the recessive alleles. When we cross it with that dihybrid female, she has a variety, remember, of gametes that can be produced. But remember, during meiosis 1, we do have some crossing over occur. So that's indicated by the different colors that you see here. So remember, crossing over is simply when you have those sister chromatids that produce that tetrad, all right, during prophase 1. Sometimes those chromosomes from those homologous pairs are going to basically kind of become intertwined with each other. And when they do that, they can actually switch parts. So those genes can actually switch positions. And when that occurs, you get some recombinant chromosomes being produced. And so that's going to introduce that variety that we're seeing. Now, because it doesn't happen all the time, again, we don't get that typical 1:1:1:1 that maybe we would expect. What we end up getting is we end up getting a ratio that's a little bit different. So primarily, we have the, again, the parental type offspring. So again, these are the ones that resemble the parents, which is kind of what we would expect to happen. But the recombinant offspring also occur as a result of that crossing over. They don't occur in as great as numbers, again, because crossing over doesn't occur all the time, but they are present. So what we do is we take those numbers and we actually calculate something called recombination frequency. And so you basically look at the numbers that you have, in other words, the unexpected recombinant offspring numbers, you add them together, and then you divide it by the total number of offspring, and that's going to give you a frequency number. And that frequency for this particular trait in this example is going to be 17%. And that's going to be important as we move on into the next screen. Now, basically, the information that Morgan had gathered was actually looked at by another scientist, and this was actually one of Morgan's students. His name was Alfred Sturtevant, and he constructed something called a genetic linkage map. And this is basically just an ordered list of genetic loci, again, positions along a particular chromosome. He predicted that the farther apart two genes are, actually the higher the probability that a crossover will actually occur between them, and therefore the higher the recombination frequency number. So the distance between two genes is going to be expressed as a particular unit, and we call those map units. And sometimes these map units are also called centimorgans. And these units represent a 1% combination frequency. Now, these are relative distances, all right? So in other words, they're not very precise, but they do give you an idea as to where these genes are located on the chromosomes. Genes that are far apart in the same chromosome can have a recombination frequency near 50%. And when you have something pretty close to this number, they may be physically linked but genetically unlinked. In other words, they're going to behave as if they are found on different chromosomes. And so this kind of gives you an idea of what Sturtevant was looking at, or sort of hypothesizing based on the information provided by Morgan. And so if you notice, these genes right here, we have a B and we have the VG, so this would be the color, and this is going to be the type of wing. If you notice, we think about them, remember we had looked at that number where we calculated the recombinant frequency of 17%? We might subjectively, in other words relatively, put those particular genes at about these positions on the chromosome. And then if you look at this gene, again, I can't tell you exactly what it is because I'm not quite sure, but there might be a situation where this one, based on the relative distance that we have from those recombinant frequencies that we've calculated, might actually end up between the two that you see right here. And you're going to get some practice on this in class, and we're going to look at some different situations where you're going to have to first calculate the frequencies and actually create your own genetic linkage maps. Now, sometimes we have things occur during meiosis that kind of cause problems with what we might expect in the phenotypes that are produced in the offspring. We talked about the crossing over in the previous couple of screens, but now we're going to look at something called abnormal chromosome number. Now, when you have chromosomes that are maybe not quite the number that they should be, we know something has occurred during meiosis. We call this nondisjunction, which basically means pairs of homologous chromosomes that do not separate normally during meiosis. As a result, one gamete receives two of the same chromosome, again because of an error in meiosis, and possibly another gamete might receive no copy of that chromosome. And that's what you're noticing down here towards the bottom. This would be considered nondisjunction in this particular meiotic event. So instead, during this meiotic event, during the separation of those sister chromatids in meiosis II, instead of one going this way and one going this way, both chromatids actually went to the left. And that resulted in one of the gametes actually having more and one of the gametes having less chromosomes. And of course, that's going to affect the offspring that are going to be produced from these gametes. Now, we have something called aneuploidy, and this is going to result from the fertilization of gametes in which non-disjunction has occurred. Offspring with this condition have an abnormal number of particular chromosomes, like we had said. One group we call a monosomic zygote, and this is going to be a situation where it has only one copy of a particular chromosome. Another example would be trisomic zygote, and this is going to be one that actually has three copies of a particular chromosome. We also have a situation where instead of aneuploidy, you have something called polyploidy, and this is going to be a condition where the organism has more than two complete sets of chromosomes. Triploidy, represented by 3N, is 3 sets. Tetraploidy is going to be 4 sets. And actually, polyploidy isn't necessarily a bad thing, but it is more common in plants than it would be in animals. Now, in addition to an abnormal number of chromosomes, we might also actually have breakage of a chromosome, and this can lead to 4 types of changes in chromosomes. One of those being deletion. In other words, you simply remove a piece of the chromosome. So in this case, we have letter D that was removed from this chromosome. So obviously, we're going to be missing genetic information. We have duplication. In other words, we have a particular gene that's been duplicated. And so when you look at that, sometimes that can be a bad thing, believe it or not, that we actually have more information than we're supposed to. Sometimes not, but sometimes it can. So then what we need to do is we need to talk about something called an inversion. And an inversion is a reverse of a segment within a chromosome. So if you look over here at letter C, you're going to notice that this is going to represent our inversion. And if you look at the set of genes that you see right here on this chromosome, you're going to notice instead of B, C, and D over here for the inversion, we have D, C, and B. So they've basically been kind of reversed in their arrangement on the chromosome. The information is still there, but sometimes the way the information is being read could be somewhat dangerous or detrimental to the organism. Now, the fourth one is something called a translocation, and this is going to be the movement of a segment from one chromosome to another. And so letter D represents a translocation. And so if you notice, we have two different chromosomes here. And so what we're seeing is we're actually seeing the movement of genes on this chromosome over to the one that you see below, then of course the movement of genes on that chromosome to the one that you see above. And so this would represent the translocation idea of some alterations to chromosome structure. So some disorders that are actually due to chromosomal alterations, one of those would be Down syndrome, and this is going to be an aneuploidy condition, and this is going to result from 3 copies of chromosome number 21, and there's actually different varieties of Down syndrome or different variants of Down syndrome, and actually some of them do not have 3 complete copies of chromosome number 21. Sometimes there can be bits and pieces of that chromosome. Down here towards the bottom, what you see is something called a karyotype, and so sometimes when we have offspring, a baby that actually is it's suspected to have Down syndrome, they will do a karyotype of that individual to determine whether or not there are extra copies of chromosome number 21. So what we looked at in the previous screen is the idea of this whole idea of aneuploidy in autosomes, but there can also be aneuploidy in sex chromosomes. Now, when you look at aneuploidy in sex chromosomes, this can produce a variety of conditions. One of those being something called Klinefelter syndrome. So instead of just simply having XY, you have somebody that actually has XXY. So this is a result of an extra chromosome, an extra X, and this produces individuals with this particular disorder. In females, you can have something called monosomy X, sometimes called Turner syndrome, and this is actually the only known viable monosomy in humans, but instead of the female having XX, the female actually only has one X. And females who have this particular condition do tend to be sterile. Some disorders that are caused by structurally altered chromosomes, which are the ones where you have either deletion, inversion, etc. One of those would be cry of the cat, and this occurs because of a deletion in chromosome number 5. A child who is born with this syndrome, it would be mentally disabled and has a cat-like cries. So that's one of the early indicators that there's probably something wrong with that particular baby. Unfortunately, individuals usually die in infancy or early childhood. Certain cancers can also be caused by structurally altered chromosomes. One of those would be CML, and these are caused by translocations of chromosomes, so the exchange of different genes from different chromosomes. Alright, so that's going to finish up this screencast. I know this one might be a little bit longer, but I didn't think we needed to break this one down into 2. As always, please remember to make sure you have completed your screencast study guide.